Good morning.
Morning.
So Norman van Rhijn is a PhD candidate at the University of Manchester, and his presentation title for today is Fungal Shapeshifting Meets Drug Resistance. Norman is a research associate at the Manchester Fungal Infection Group at the University of Manchester, and after obtaining his bachelor's at Leiden University, he moved to Manchester for a master in mycology at the University Hospital of South Manchester. He did his PhD at the University of Manchester in molecular biology, and his main interest is the relevance of fungal fitness with a focus on Aspergillus fumigatus, I hope I pronounced that right, and different growth phenotypes in pathogenesis and drug resistance. By using large-scale screening methodologies paired with optimized CRISPR-Cas9 protocols, large libraries of fungal isolates can be screened for a wide range of characteristics. Thank you for joining us today, Norman. I'm looking forward to your presentation.
Hopefully that works now. All right, so first I want to thank Ido and Danny for organizing this excellent online conference. I'm really enjoying it so far, and the diversity of the talks has been amazing. Today I want to talk to you a bit about fungi and hopefully teach you a bit about the world of fungi. There we go. Most of you, when you think about fungi, think about something like this on the left, mushrooms, the things you eat in the kitchen. But talking about mushrooms as fungi is the same as talking about plants and just thinking about an orange. Both of these are the fruiting body of the organism. Now, for plants, we have about 400,000 species, and 99% of these have been described. However, for fungi, we've got about 3 million species. This is an average because we're not sure, because only 5% of these 3 million have been described. Now, fungi are very important for our lives. The classic example that we have is the penicillin. It's made by a fungus, but a lot of other things are made by fungi, statins and cyclosporin, anti-cancerous compounds, antivirals. We can't drink our favorite drinks without fungi, the citric acid in most of our fizzy drinks. And of course, beer is partly due to fungus. Our bread, yeast, is a fungus. And nowadays, a lot of building materials and also cleaning up of oil has been tried with fungi. However, there's also a dark side to fungi. Fungi infect a lot of animals on our planet. So amphibians, mainly our small frogs, are very prone to fungal infections. Penguins, chickens, and this cute little bird on the top left, which is a cockapoo, which is actually a quite endangered species, but they're very prone to fungal infections. Also, a lot of plants on the world are quite sensitive to fungal infections. On the bottom, we see a rice plant with fungal infection, rice blast fungus, potatoes, and I think, as we're going to hear later, a little bit about bananas as well. But what we focus on in the lab is fungal infections of humans. And mainly in my research, I'm focusing on Aspergillus. So on the bottom is how it normally grows on a plate, on a petri dish. It grows as a little, well, as you probably know from when it grows on your food, it grows quite fluffy and colorful. And then when we look at it under a scanning electron microscopy, it forms these long filaments, and then the fruiting body, so what we in larger fungi would see as a mushroom, this sort of head with these little balls on top. These balls are the spores. They can easily fly away with a little bit of wind. So you can already see that half of these have been blown away. And these spores are usually what flies through the air. Loads of them are in the air, and you breathe them in, probably 100 a day. But normal people can cope with these fungal spores and clear them due to a sufficient immune system. However, we get more and more people that are immunocompromised. And that is due to progression in medicine. Over the years, we get more and more sophisticated methods of treatments. So for example, organ transplants, people longer in ICU, cancer treatments and stuff like that. Now paired with that, we have an increase in resistance to the gold standard, which is an antifungal family called the azoles. We see a massive increase in resistance to these compounds. So it's a double problem, and we're working on new antifungals and modes of resistance to these antifungals. Now I wanted to open with a little bit of an anecdote. When I first started my PhD that's 5 years ago now, we wanted to barcode lots of fungal isolates, and each one is missing one genetic regulator. So we wanted to put in a piece of genetic code so we could recognize them on a sequencer. Now this all worked fine until I got to the sequencer, and stupid enough, I broke the sequencer. Now, for you, people that work with sequencers, a sequencer is about $128,000. So breaking one wasn't very clever. And we had to find other ways of actually doing our analyses. Now the second bit was, my supervisor said, you know what, we're just going to do it lower throughput, grow it in a shaking incubator, as you see here on the top, and you just take samples every 2 hours for 48 hours. Now, as you can imagine, that meant staying overnight in the lab, so I had one of these wonderful sleeping bags underneath my desk. But what we didn't think about is that I had to keep the shaking incubator open for about 30 minutes every 2 hours, and that affected our readings. So I played with these two things for about 1.5 years, and actually in the end of my PhD, nothing that I did during this 1.5 years ended up being in my thesis. So just to take away messages, experiments will fail and it's not always your fault, but hopefully, as I'll show you, that it was all for the better. So after this 1.5 years, we decided, you know what, let's just go completely low throughput. We'll go one by one and just look under the microscope for how it grows under different conditions. And then you just have to do it by eye and measure it all by hand, which is a lot of work, but thinking that I already lost 1.5 years, I agreed to this. So as you can see on the top left, this is what the start of what our fungus grows like when we look at it under the microscope. So this is a GFP labeled fungus, and that's just from the spore, this large big blob with, well, I would almost say 2 eyes, it looks like it. Starts to grow, its filament, and then we can follow that in time in 3D and do a lot of quantification. But as you look at the population on the right, you can see that there's a lot of different stages. So a lot of these fungal spores have already grown long filaments, some of them have grown short filaments, and some of them just clump together. And that's what sparked my interest in doing a bit of, well, actually more population dynamics. But the first output we got was we screened 500 of these isolates that missed one genetic regulator, and we measured their sort of fitness under these drugs and tried to find strains that are resistant to this drug, itraconazole. And our main interest was this strain missing a genetic regulator that we called NCTA. Now this is one that wasn't described yet in the literature, so that's why we looked into it. And then when we actually looked at different characteristics of this strain, we found on both of these top things, so these are epithelial cells, which is normally the cell that it interacts with first in our lung, and macrophages, which normally clear up the fungus. It wasn't able to cause damage, in the damages in red for this particular strain. It wasn't able to cause as much damage as the normal wild type strain. However, when we looked at immunological markers, these are sort of the messengers of the immune system, we saw a massive increase in these cytokines, which meant that the immune system recognized it more, but the fungus wasn't able to cause any damage. Now, normally when we see something like this, it means that the outer structure of the fungus is completely different, which was something I couldn't pick up with a sequencing machine, but actually under the microscope worked out quite well. So in the end saved this paper. And then when we looked under a transmission electron microscope, at the top is what you normally see of a spore, so that's the wild type, but at the bottom we saw these sort of, well, we're still unsure what to call them, but we've called them fungal hills on the outside, and this is probably what is driving this immune response, but we're still characterizing this because we have no clue what these are. Now, the second story that I want to talk about is we also looked at another drug which is called Congo Red. Congo Red is normally a dye for clothing, and it's also used, as you can see here in the bottom left, as a dye for histological sections. So anything that's red is this Congo Red dye. Now, traditionally, Congo Red, if you screen for fungal resistance to this, you do this on plates. As you can see, the more red, the higher the concentration. And we found 2 isolates which are hypersensitive to this drug, and 2 isolates which are resistant to this drug. But as I previously had such success with just looking at the microscope, I thought, you know what, maybe it's more interesting to look under the microscope instead of just doing it on plates or doing it in another fancy way. And actually, this proved to be successful as well. We found that this fungus normally grows these spores that you can see on the top. They're quite small, but when we add in the drug, they swell up to about 1,000% of their normal size. And these are not all the spores, so it's a subpopulation of the spores again that take up these drugs, form massive storage units, and allow the other spores to actually grow. Now, normally when we see something like this, it means that it's an uptake difference. So we characterized uptake of this Congo Red, looked at the half-life, and we saw massive differences in these strains. So actually the hypersensitive ones are the ones that do not take up this Congo Red, and the resistant ones take it up more and therefore allow fungal growth. So the main message here is, if some experiments don't work out and you think I have to go back to the drawing board, sometimes that's a good thing, and you probably find something more interesting than you initially would have found. Now, what are we doing? What are other things that we are doing? We're looking at resistance to the new antifungals, and actually we're looking under the microscope again, so I convinced my PIs that that's the way forward and not something more fancy. But on the side, I'm working on new genetic tools, some high throughput screenings, so trying to optimize this microscopy and doing a bit more genetic manipulations via CRISPR. Another story that we're working on is antifungal loaded spider silk. So this is a collaboration with Jolanta from Nottingham, and we met on Twitter via one of these, well, kind of interactive seminars, and we decided to do something together. So maybe for one of you from this conference, something can come out of it. But my main project is I'm now looking at development of resistance. So in agriculture, we use a lot of antifungals. We see hotspots of resistant fungi in compost heaps. So I'm now starting to look at the evolution of pathogenicity and actually going back again to doing a lot of microscopy, which I just love. And I would like to thank all these people, my supervisors from the Manchester Fungal Infection Group and the University of Exeter, and our collaborators from Imperial College London, the Bioinformatics Unit and Institut Pasteur in Paris, of course our funders. And if you've got any ideas, want to collaborate, or you've got any questions, just send me an email or a message on Twitter, and I'll always reply.